Today, we're diving into the world of microbiology. And let me tell you, it's a world where scientists are fighting a war on a scale so small you can't even see it. In this invisible world, control is everything, because before you can even start an experiment, you have to build the perfect battlefield.
So think about it. When you're dealing with tiny organisms like bacteria, how do you make sure you're only studying the ones you want to study and not the millions of others just hanging out in the air? Well, it's a two-part answer. First, you have to completely wipe the slate clean, and then you have to find a way to grow your evidence that you can actually trust.
So let's get into part one, making things perfectly, absolutely 100% clean. In a lab, anything and everything can be a source of contamination. The only solution is, well, total annihilation. And one of the best ways to do that is with something surprisingly simple: scorching dry heat. The technique is called dry heat sterilization, and it is a total brute force method. We're talking about basically a super-powered oven that blasts things with incredibly high temperatures for hours at a time. The goal? To completely destroy every single trace of microbial life. No ifs, ands, or buts. There are no survivors.
Now, you might be thinking, hey, why not just use steam? And that's a great question. That's called moist heat sterilization. And yeah, it's definitely faster, but it's not the right tool for every job. You can't exactly steam clean things like powders or oils or certain kinds of delicate metal equipment. For those, dry heat is the only way to go. Sure, it takes a little longer, but it gets the job done without ruining your stuff. So how does it really work? Well, it all comes down to a process called conduction. First, the oven heats up the outside of whatever is in there, let's say a glass Petri dish. Then, that heat gets absorbed and starts to travel from the surface right down to the very core, until every single part of that dish hits a temperature that microbes just can't survive.
And this isn't guesswork, it's an exact science. There's this really fascinating trade-off between temperature and time. If you crank the oven up to a blistering 170 degrees Celsius, you only need about 30 minutes. But if you drop it down to 150 degrees, you're going to need to wait 2.5 hours. It's a very, very specific recipe to get that perfectly sterile result.
So what kind of stuff are we talking about putting in these high-tech ovens? All the essentials. Think glassware, like Petri dishes and flasks, or delicate powders that absolutely can't get wet, oily substances, and of course, super sharp metal tools like scalpels and scissors. All right, so we've done it. We've created a perfectly sterile environment. Our battlefield is clean. But here's the thing. That pristine setup is totally useless if the way we grow and count our microbes is flawed. And that brings us to our second big challenge, which hides inside a classic technique called the pour plate method. So here's the rundown on how the pour plate method works. You take your liquid sample, put it in an empty Petri dish. Then you pour this warm, nutrient-packed jelly, it's called agar, right over the top. The agar cools and solidifies, trapping the microbes inside, and they start to grow into little colonies that you can actually see and count. I mean, this is a standard method. It's used in labs all over the world for checking everything from food to cosmetics to medicine. It sounds pretty straightforward, right? So what could possibly be the problem? Well, as it turns out, there are a few pretty big catches that can really mess up your results. Okay, here are the hidden flaws. First off, that molten agar is kept at around 48 degrees Celsius. That's hot enough to actually kill some of the more sensitive microbes you're trying to grow. But if you let it get too cool, it turns into a lumpy, useless mess. On top of that, when you sterilize the agar and the nutrients all together, they can react and create toxic stuff that stops bacteria from growing at all. And to top it all off, trying to count little colonies stuck inside a cloudy jelly, it's just really, really hard.
So with all those issues, you'd think the solution would have to be some super complex, high-tech thing, right? Nope. Turns out a recent study found a fix that is so simple it's brilliant. It addresses all of these problems at once. It's a true aha moment for a technique that's been around for decades.
Just look at this comparison. The old way, you mix everything together, sterilize it, and you use about 15 grams of agar for every liter. The new way, you sterilize the nutrients and the agar separately. Then you just mix them together right before you pour. And get this, you also use a third less agar, only 10 grams per liter. That's it. 2 tiny little changes. So, the big question, does it actually work? Oh yeah, it works. Let's look at the numbers. This first chart is for Staphylococcus aureus. With the standard method, they counted on average 89 colonies. But with the new modified method, That number shot up to 118. That is a 32% increase. That is a massive jump in accuracy, all from that one simple tweak.
And it wasn't a fluke. They ran the test again with a totally different microbe, E. coli, in a different medium. The results? Even more dramatic. The new method gave them almost 38% more colonies. This simple change is consistently giving a much more accurate picture of what's really in the sample.
I mean, you don't even have to take my word for it. The researchers who discovered this summed it up perfectly. They said, separating the agar from the other components and reducing the agar concentration can enhance microbial growth. It's a direct confirmation of everything we just saw. So simple, so effective. So what's the winning formula here? Why is this so much better? Well, first, by sterilizing separately, you don't create those toxic compounds that kill the microbes. Second, using less agar makes the jelly softer, which might let more oxygen and nutrients get to the microbes. It also makes the colonies way easier to see and count. And here's a great bonus. Labs can now keep one big batch of sterile agar ready to go and just mix in different sterile nutrients when they need them. That saves a ton of time and money. Okay, so we've been deep in the weeds of lab procedures, but why does this level of precision really matter to any of us? How does a 30-something% improvement in a Petri dish actually affect our daily lives? Well, the answer is a lot more than you might think.
The big idea here is that in science, the tiniest details can have the biggest impact. The difference between a result that's just acceptable and one that is truly trustworthy, well, it often comes down to small changes in procedure, just like the ones we've talked about today. It's all about getting a more honest answer.
And that trustworthy answer has a huge real-world impact. It means getting more accurate counts of harmful bacteria like Salmonella in our food. It means making sure the cosmetics and medicines we use are free from contamination. And it builds a more reliable foundation for all kinds of future research. Simply put, better methods lead to safer products for every single one of us.
And all of this leaves us with one final thought. You know, we often think of scientific breakthroughs as these huge, complex, brand new inventions. But as we saw today, sometimes the biggest leaps forward come from just taking a fresh look at the tools we already have. It really makes you wonder, doesn't it? What other simple changes are just sitting there, waiting to be discovered?